So let's look at section B now of the 2018 paper with module 1 question enzymes with reference to the structure of enzymes. Distinguish between competitive and non-competitive inhibition. Now, what is competitive inhibition? What does that speak to?
Um, Basically, those are inhibitors that have similar structures to that of the substrate in which the enzyme would connect to.
Okay, competing.
They also bind reversibly to the enzyme active site.
All right, so binds the active site, reversibly to the active site. All right, so we're looking at that. They find that they bind reversibly. Um, so the substrate in question, the inhibitor, right? We know that it has a similar structure, structure, right, to the desired or the suitable substrate, and they bind what you said. No, they bind reversibly, right? So we have that there, similar, right? So looking at that, they're competing with the active site there. And when we talk about non-compet now, non-competitive inhibition now.
This
They're
is
going to talk about it
when
bind
a
to.
substance binds to an allosteric
They're going to talk
site.
about it bind to.
Okay.
Allosteric site, that's not the active site. It's anywhere but the active site.
All
But
right.
it can change the active site. So the substrate cannot interact with.
Okay, lovely. Binds to the allosteric site, to the allosteric site, right? And that will change the structure, the tertiary structure of the enzyme, the tertiary, right, structure of the enzyme, and that would speak specifically to the active site. Yeah, change the structure of the active site. Yeah. All right. Lovely. So you're just going to be discussing that different structure of enzymes, tertiary structure of enzymes, and stuff like that, the compatibility and stuff like that, right? And generally, an example would have been in. Oh my God! Did I just forget the structure? I forget the name of the thing. It's a diethyl mm, 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 toluamide, right? And diethyl toluamide. Hopefully that's the compound. You guys can look it up, right? Which actually affects acetylcholinesterase, right? And stuff. We usually speak about the example for competitive and non-competitive inhibition with that compound, right? And how it is used in DEET, right? It's DEET, right? And in diethyl, right? Toluamide, right? So it's DEET, right? So looking at those type of stuff and how it is used within insect repellents and how it actually neutralizes acetylcholinesterase within enzymes and within the insects, right? And actually provide for repellents using inhibition. So the thing is, they might not ask you about the stuff specifically. They might give you a scenario and ask you to use your knowledge of inhibition, right, and stuff like that to now describe something. You guys should be able to do that. Discuss the role of four tissues found in the root of the plant to support major functions of the roots. What are the major functions of the roots?
Start with the cortex, the one.
And the cortex contains what type of cells?
So the parenchyma.
The parenchyma. Okay, let's speak about this, right? The exodermis, right, is the sclerenchyma, right? Um, I'm gonna fix the spellings, right? The cortex is the parenchyma, right? I spelled sclerenchyma wrong. Oh yeah. Alright. So exodermis, right, and the cortex, sclerenchyma. Right, we'll also have some people can just speak about the xylem and the phloem. as well. Right. We know the flow of water throughout these things. So we know that the exodermis specifically, right, or the epidermis, right, we'll cover the exodermis and we're gonna have projections of sclerenchyma, right, within the walls of the root, producing root hairs. So you could speak about the role of the exodermis as it lets in water, right? And the endodermis has the fact that it can actually let in water into the root. The parenchyma has the osmotic gradient, right, set up, right? So we can actually have the movement of water towards the vascular bundle. We know the xylem itself will contain the high concentration of minerals. Therefore, that is what set up the osmotic gradient, right? We know the flowing would facilitate the movement of nutrients towards the root cells and specifically, while the xylem will actually facilitate the movement of the water and minerals up through the roots towards the rest of the plant. All we're doing is really describing the role of four tissues. That's all we're doing. They're gonna speak about the root. You could speak about the root hairs increasing the surface area, right? That could actually help with the function of the root. One function is uptake of water, right? We could be speaking about those things and how it really ties into the main function, right? So the main thing is to uptake water, right? The sclerenchyma and the parenchyma help set up a gradient so the water can move through. The sclerenchyma has projections, right, called root hairs, which increase surface area. The xylem sets up the osmotic gradient generally, right, and transports water from the root to the rest of the plant. All of these speak about that. The phloem produces a supply of nutrients that will help in the proliferation and the health of the root. Speak about these things and how it ties into the function of the root. That is all that we must do. Alright? That is all that we must do here. And then we head into the module 2 section, right, of the section B for this paper.